Well, thanks everybody for coming out so bright and early to talk about structural variation. I'm going to start with the genome, and if you've seen this slide before, don't give it away. But so this is a staining of a human genome, and the question is, just by looking at this human genome and the staining of it, can you tell anything about this person? You know, could you make a diagnosis, say, or tell me something about this person? Yes. Yes, if you look at the bottom down here, there's actually 3 little chromosomes down where it's labeled 21. So this person has trisomy 21. They have Down syndrome. And here's another example. This is a different human genome, but it also has some things you can notice just by looking. Do we have anybody from the audience? What can we see by looking at this genome? 7 and 8. Yeah, there's extra 7s and 8s, I guess. We have a particularly large 8. We've got little arrows to help you. So some chromosomes, 11's missing a chromosome arm. So this is a cancer genome. It's pretty typical in many kinds of cancer. You get gains and losses of huge segments of DNA. And here's a last one. This one's a little trickier. But is there anybody who knows what chromosomal abnormality this is? Yes, this is the so-called Philadelphia chromosome. I think it dates from around like 1960 it was first described, and it's a recurrent translocation that happens when part of chromosome 22 gets fused to chromosome 9. And it turns out that 95% of a certain kind of leukemia, CML, has this recurrent translocation, and about 25 to 30% of adult onset ALL, I believe, also has this recurrent mutation. So these kind of gross chromosomal abnormalities you can see from these stainings of people's genomes, but these you can only really see things here at this megabase and chromosomal arm sort of scale, and we didn't really have a good way to look at abnormalities on a somewhat smaller scale until some technologies came along, like comparative genomic hybridization, where the idea is here that you could take 2 DNA samples and you could label the 2 different samples differently, one with a green dye, one with a red dye. And then if you can put them the 2 samples side by side, differentially labeled, you can compare the colors to compare the abundance of the DNA of each kind. So here we have a tumor and a normal sample, and we can use this normal karyotype as kind of our bait. And you can see that there's different colors of the red and the green, so you can get at, for example, a cancer genome, and you can see the gains and losses by the relative abundance of the color, which is telling you about the relative abundance of the DNA. And then this was kind of systematized and automated by using microarrays. So then we get array CGH. But it's the same principle. We take 2 DNA samples, a tumor and a normal, or 2 healthy individuals, and you can see the relative change in the DNA abundance depending on the different baits you're using on the array. And this works to find CNVs at resolutions of megabase scale, maybe hundreds of kb. And it's become kind of a staple of clinical diagnosis in certain cases, especially children with developmental delay, or other problems that show up in childhood, maybe autism. And although interpretation in some cases remains hard, we can see large chromosomal abnormalities with this kind of technology. And this is, as far as I know, kind of the gold standard against which other things would be measured today, especially for clinical relevance. And then if we go forward a few years, right, arrays were beginning to be used to do GWAS studies. We were making a lot of SNP arrays, putting more and more SNPs on them. And people realized that in addition to using the arrays to just detect SNPs, many of these arrays work by, there's 2 probes on the array, one to detect each allele of the SNP. And then in the same way sort of that CGH works, you get 2 differential, maybe fluorescent signals off of those 2 probes. But in addition to just looking at the difference between those probes to interrogate the SNP, you can also look at the intensity of the probes together. And this also tells you about the abundance of the DNA. And this is a little bit of a noisy signal if you look at one probe. But if you look at many probes across a stretch of the genome, you can begin to see patterns that tell you if there's more or less DNA. So people discovered that you could do copy number analysis using the data from the SNP arrays. And in at least one case, in the AFI 6.0, in some work done at the Broad, we actually added copy number probes to the AFI 6 array, especially in some regions of common CNVs, to get a better measurement of copy number from the array data. So right along with your GWAS, you could also get copy number readout. This tends still to target CNVs that are in the 50 to 100 KB range and up, something like that. But this was a big advance in being able to detect slightly smaller CNVs. And then kind of the surprising thing that was found was that there was actually widespread copy number variation. These large chromosomal abnormalities you don't see very much. They tend to be associated with really extreme phenotypes. But we began to discover that there were really common CNVs, even of this order of several hundred kilobases, in the average healthy population. But of course, today we live in the era of sequencing data. So now we don't so much just have arrays anymore, but we have lots of short reads. And so people wanted to begin to interrogate structural variation and copy number variation from these short reads. And so the problem is really about ascertaining these very large variants from the very tiny reads. And so there's basically three signals that people look for in short read data like Illumina data. And the first is kind of a signal from read pairs. When you have paired end data, you typically know the target fragment length that the pairs are coming from. Maybe it's, say, 400 base pairs. And so if you take a pair and align it back to the genome, you expect it'll align about 400 base pairs apart on the genome. But if that read pair happens to span a large deletion, say, then what happens is the pair will align further apart on the genome than you would expect, because the fragment's only 400 base pairs, but the reference is longer because the reference has extra DNA compared to the sample you're interrogating. And if there was an insertion of sequence that was not on the reference, you might see one end of the pair align, but the other one doesn't, because there's no place for it to go on the reference. Or if there was a tandem duplication, you might see that the pair now is in the incorrect orientation, because this read pair is really on a duplicated stretch of DNA and maybe spanning a join that's not on the reference. And so when it maps back to the reference, it looks unusual. The second major signal that people look at in short read data is just the number of fragments. So this is very similar to the idea with looking at the intensity in the SNP arrays, just that the number of fragments is going to correlate with the abundance of the DNA. It's a little bit of a noisy signal again, but when you look at this abundance over stretches of the genome, you can see if the DNA is over or underrepresented, and you can infer deletions and duplications. And the last signal people look at is split reads. Especially if you have longer reads, you might get very lucky, and the read might actually span a deletion or a duplication. So if it spans a deletion, for example, you'll see two ends of the read, of the same read, that are aligning far apart on the genome. And for duplications, you might see where they kind of wrap around. And essentially, these reads are telling you about junctions between two pieces of DNA where there is no junction on the reference. And you don't always get a split read signal if there happens to be a lot of repetitive DNA where this junction is happening. You might not be able to detect that signal. But these three signals are generally what most methods are looking for in this short read data to try to infer structural variation. So one variation on this theme is to look at these signals in exome data. And as exome data began to become widely available, people began to develop methods to try to ascertain copy number variation from the exome data. And one of the difficulties with using exome data is that the capture process introduces a lot of technical noise. So if you just look at the number of reads over each target, so the way the capture process works is we have certain baits that are pulling down the DNA in certain target regions. So you have a bunch of targets, usually corresponding to an exon. And then you can look at the abundance of those reads, but the capture process, because it's noisy, you get a lot of variation. So there's different techniques people use. Some work by Menachem Fromer used removal of some of the first principal components to try to reduce the noise. And in general, you can call CNVs from exome data, but usually you need 2 or 3 targets in a row to get an accurate call. And you can usually tell if there's extra DNA or less DNA, but you may have a little bit more difficulty quantifying how much extra DNA there is or how much depleted DNA. But there are methods out there for calling CNVs from exome data. And then for whole genome data, there are a number of methods. One that we worked on in our lab is called genome strip. And in the 1,000 Genomes project, it was probably the most accurate method for calling structural variation, especially in the early phases of the 1,000 Genomes project. And what made the method unique is that it was really one of the first methods to both combine all three of those signals I talked about, the split reads, the read pairs, and the read depth together at the same time in one form or another. And then also to combine that with calling across multiple samples. So rather than looking at one person at a time, you look across many, many people. And both of these turned out to be really important. If you look at the history of development of methods in this area, methods that used 2 different signals or more tend to be much more accurate than methods that are using just one, because there are technical artifacts that show up that will confound one method. But when you get concordance between 2 different signals, you get a much more accurate result. And also looking across multiple people helps you be accurate because, for example, if you see a little bit of evidence for a CNV, but that evidence is spread across just about everybody, it's much more likely to be a technical artifact. A true CNV, you'll see it in some people, but not in others, because this is a real genetic variant, so it should be following population demographics. So when these methods were applied in the first phase of 1,000 Genomes back in around 2011, the project mostly focused on calling on deletions and mobile element insertions. And this gives you some idea of the abundance of things we found. We found, thousands of common deletions and polymorphic mobile elements like this alu peak shown here. And so this was kind of the state of the art in 2011, but it created a reference of many, many people from different ethnic backgrounds and some glimpse of the abundance of this kind of variation, and also the kind of size range we were seeing, although there's going to be a certain amount of under ascertainment here because 1,000 Genomes was all low coverage sequencing.
You're still at the 50K resolution?
No. So this is going in resolution down here to well under a kilobase. That large green peak there corresponds to alu transposable elements, which are active in the human genome, and which I'm going to talk about more in a second. But we're going down well below 1 kb here. We're probably losing power somewhere below 1 kb. So a little later, I'm going to show you probably a more accurate assessment of the actual length distribution. I guess the gross thing to take away is that as there are fewer large variants we find in the population, right? We see in healthy people occasionally megabase scale deletions and duplications, but not very many. There are some very common things at the 100 of kilobase scale, and as you begin to go smaller and smaller down to like the 1 kb length scale, there become many more CNVs at all different frequencies that are in the human population. And this continues right down to indels, right? A typical genome might have, a couple 100,000 indels, but 3 million SNPs. So as the variant gets larger, in general, the frequency gets lower, except for things where there's a specific mechanistic cause, like this alu insertion here.
At what point do you run into triplet repeats of normals?
So at what point?
What resolution size? Where you get triplet repeats that vary in normals? Fragile X, Huntington's, there's a range of normal variation in the triplet repeats.
So how big do those repeat expansions get, I guess is kind of your question. I mean, I'd refer you to Melissa Gimrich's recent paper, which I almost put in a slide about here, except she's really working on these tiny things, which are these short tandem repeats of up to 6 mers, but which then may repeat many times. Like I think it's not unusual to get like 100, but still you get 100 times 6, you only have 600. Is that really structural variation? I decided not to put that in this talk, but I think I would look at that as kind of the latest sort of definitive reference on the range and amount of that kind of variation. I think my sense is that most of it tends to be under a couple 300 bases. You don't get, of things that long, you don't get so many places in the genome where the repeat count gets that high. But that is another form of variation that in many ways shares features that are similar to these larger scale structural variants. So another class of variation that gets lumped in with structural variation is caused by retrotransposons that are active in the human lineage. These are mostly ALUs are the most common, and then L1s are a little less common, and there's also SVAs that are a little bit more rare. So ALUs are about 300 base pairs long, and when they insert in the human genome, so that they're a copy and paste sort of retrotransposon, so the families, especially the ALU-Y family, which is active in humans, will insert new copies into the genome at some frequency, and then these will become polymorphic in the germline. And ALUs are about 300 base pairs long, and most ALU insertions are full length. So when you see a new ALU show up, it'll usually be about 300 base pairs, and depending on the family or if it's mutated, it'll then remain active, and more copies will show up in the genome. And so when you go to detect these in the human genome, you can see them either as deletions relative to the reference, meaning that the reference has captured the ALU in the person that the reference genome was taken from. But then some people may not have that ALU because it'll be at some frequency in the population, so it'll look like a deletion relative to the reference. Or if the reference didn't capture the ALU, especially if it's more rare in the population, you'll see it as an insertion relative to the reference. And generally the methods for finding these are a little bit different. Because ALUs and L1s are so common, a couple people have written methods specifically to target this kind of variation. Chip Stewart wrote the first method that I know well anyway, and it was a great method for doing this when he was at Boston College, and he's now at the Broad. And there's a new method. Chip's code sort of fell into disrepair a little bit in the meantime after he left and came to Broad, but there's a new method by the Gardner lab at the University of Maryland called MELT, which in 1,000 genomes has looked like it's a pretty accurate method. So that's mostly, I think, what I was going to say about ALUs and L1s. Oh, I suppose the other thing is I mentioned that ALUs are mostly full length. When L1s insert, it's quite common that they're not full length. So a full length L1 is about 6 kb, but often you'll see L1 insertions that are not full length. Now, if they're not full length, they can't then reinsert, but you'll get these L1 insertions that are shorter than 6 kb, which are fairly common. So far I've talked about CNVs that are mostly fairly simple. They may be fairly large stretches of DNA, they may have to do with retrotransposons, but they're all kind of this traditional simple model of we've got just a deletion of a little section of DNA, or we've got a duplication, so we have one extra copy. But something we've been learning over the last few years is that structural variation can be a lot more complex, at least in some number of cases. And there have been hints for this for a long time. When people go in and look at deletions and duplications, they would occasionally see regions of the genome where the signals seemed to be noisier. And there were sort of hints that there were maybe many different patterns going on. And so a lot of what I'm going to talk about in the next section is just different forms of complexity that show up that don't follow these kind of naive, simple models, although there's a lot of structural variation that does. There are a lot of just biallelic deletions that are polymorphic in the human population, but not every form of structural variation looks like that. So for example, one piece of work that our lab has focused on a lot is looking at these multi-allelic CNVs. So rather than just a simple duplication, it turns out there's a lot of regions in the genome where people have 3, 4, 5, 6 copies, even 12, 15 copies of a certain segment of DNA. And that's kind of illustrated in this cartoon on the top. And on the bottom, what you're seeing here is copy number measurements we've made in 1,000 people. And the colors are showing you like whether or not the person has copy number 2 or copy number 3 or copy number 4. And you can see that at many of these loci, there are wide variation in the number of copies. And there's actually thousands of multi-allelic loci like this in the human genome. And I also wanted to mention a technology that has been very helpful in the ascertainment of this kind of variation is this droplet digital PCR. And the technology uses these nanoliter-sized droplets in an emulsion where you basically take the DNA and you dilute it down a lot so that there just isn't much DNA, so that most of your droplets have at most one copy of the piece of DNA you want to interrogate. Maybe a few droplets have two. A lot of them have zero, but we don't really care about the droplets that have zero. And then you do the same sort of labeling of the DNA, similar to the CGH, but you carry out the same reaction in every droplet. And so some droplets will just be green, some droplets will just be red, some droplets will be green and red, but you can count the abundance of the droplets, and this tells you about the relative abundance of the DNA. And because you're doing this over a whole bunch of droplets, you get very accurate copy number measurements that you can then quantize and make discrete. And this is really the only widely accessible wet lab technology like this that I know of that can give you really good accuracy for high copy number states. So this is an example of a locus that goes up to 10, 14 copies in different people. And so we can compare our measurements from sequencing data with the ddPCR measurements. And this, in our work anyway, helped reinforce that we were getting accurate results from both of them. And I want to talk about a couple things we learned about structural variation in the course of looking at these multi-allelic CNVs. And one was that there's a lot of them that are multi-allelic, and that they in fact hit a lot of genes. So you can see here that we found 1,300 multi-allelic CNVs. That's the green in the top bar over at the right. But if you look at the multi-allelic CNVs, they were more likely to be genic, and they were more likely to affect many genes. This was partly because they tended to be a little bit larger. But the multi-allelic ones actually had kind of a disproportional effect on genes. And then even more striking was the bottom bar. If you just ask, on average, take two random people, how many times do they have a different number of copies of a given gene? That signal is driven most of that signal is driven by these multi-allelic CNVs that are highly polymorphic in the population. Something like 7 times the gene dosage variation is coming from those multi-allelic CNVs. And you might also notice that very little of the signal is coming from the orange here, which is biallelic duplications. And that's kind of shown here again. It's that relative abundance of the biallelic deletions, the simple biallelic duplications, and these multi-allelic CNVs. And you can see, especially for the common variants, that the biallelic duplications almost entirely disappear. And so we began to think about why this was true. And the hypothesis here is that if you think about a multi-allelic duplication and how it forms, at one point there was probably just one copy ancestrally. And then at some point a second copy formed. But once this allele begins to go to high frequency in the population, you get a lot of chances for this allele to meet another allele that has multiple copies, and then you can get unequal crossover. When you get unequal crossover, you're going to get one chromosome with 3 copies and one chromosome with 1. And then once you get one with 3, it begins to rise in frequency in the population as long as it's tolerated, and it'll maybe meet a 2, and then it can expand again. And so the higher frequency that duplication becomes, the more likely it is to create additional high copy numbers. And another way to think about this is that being a biallelic duplication in some population genetic sense is not a very stable state, right? It tends to want to become multi-allelic or to be pruned out of the population, unlike deletion. So we see a lot of high frequency biallelic deletions, but not very many high frequency biallelic duplications. So some other forms of complexity have emerged from looking at some other technologies for interrogating the genome. Mike Talkowski's lab at MGH has done a lot of great work focusing on these jumping libraries. So unlike your normal Illumina sequencing, where you maybe have a 400 base pair fragment length, where then you're sequencing the 2 ends, Mike has specialized in making these libraries, which are more difficult to make, but they have like typically a 3.5 kilobase insert size or fragment length. So now when you're sequencing the 2 ends of the read pair, you expect them to be 3 or 4 kb apart. what this gives you for the same depth of sequencing is a much greater physical coverage of the genome. Those 3 kb fragments are spanning a much larger fraction of the genome. So if the genome is rearranged, especially in a large-scale way, you're going to see patterns of the pairs not aligning where you would think they would. And so they've been finding a lot of great things about genomes that are highly rearranged. This is showing an example of finding an inversion that's flanked by two duplications. And they're finding that while these are, a lot of these are rare, and you find a lot in sort of syndromic disease and sort of unsolved cases, that there are also places where these are more common in the germline in healthy individuals. But this has become another great technology for looking at these variations, especially on a larger scale. And sort of related to this, in the third phase of the 1,000 Genomes project, it was mostly using Illumina data, although I'm not focusing so much on the Illumina data here. But in the third phase of the 1,000 Genomes project, the Illumina methods were maturing, and people were beginning to make accurate calls in more complex variations, like inversions, for example. But one thing that people began to notice was that the inversions did not look like this simple classical model if you take a piece of DNA and just flip it in place. That often the signals looked like there was something else going on. And so using PacBio reads, we were able to validate a lot of the calls in 1,000 genomes and begin to understand what was going on. And some of the examples are shown at the bottom here. And what we're finding is that this very simple model, especially for inversions, is not actually very common. When a piece of DNA gets inverted, often it inverts to another location that's a little ways away, or it creates extra deletions and duplications at the boundaries, or it's part of some more complicated rearrangement phenomenon. So many of these loci are not this very simple model that people naively expected when they went into it. And these PacBio reads have also led to another phenomenon that's going on more and more recently, and I call it the Immaculate genomes phenomenon. And this is the idea that we can take these really long PacBio reads, which are 10 or 20 kb long, and because they're so long, we can get very good connectivity across the genome. Now, they have kind of a high intrinsic error rate, but if you have deep enough sequencing depth, you can correct those errors. And some people are experimenting with, can we use short Illumina reads to help correct the errors? And different ways of scaffolding, maybe using optical mapping. But what's emerging is a lot of people working on projects where they're assembling and making very good genome assemblies up to 28 megabase, N50 contigs, which if you're into assembly, this is really, really good. It's almost a reference quality genome that people are making from individual genomes. So there have been several efforts looking at this, mostly doing single genomes, including using this haploid hydatiform moles. There's also efforts going on right now through Genomes in a Bottle to do an Ashkenazi trio. There's an effort that's kind of come out of the 1,000 Genomes project where we're doing 3 trios, 1 Chinese, 1 Puerto Rican, and 1 Yoruban. So in all of these cases, we're making these very immaculate assemblies using these long PacBio reads and other technologies. So this is still relatively expensive to do, but once you have this very clean assembly from a single person, then it can really tell you a lot about structural variation that exists in that person. So right now it's kind of at the level where there's 10 or 20 of these, because as I said, it's still kind of expensive and a lot of work to do it. It takes a lot of sequencing with the PacBio data.
What makes them so expensive?
It's mostly the cost of the PacBio data itself, and then the fact that you have to do very deep coverage. People debate about what coverage is needed, but 40, 50, 60x coverage of these 10 to 20 kilobase reads, partly because the error rate within the reads is kind of high. You get very good long-range connectivity, but because of the high error rate, you want to cover it with enough sequencing data that you can correct all the errors. But when you do this, even on a single genome, you can begin to see in a more unbiased way than you can with Illumina data, the real extent of all the structural variation. And I especially love this plot on the upper right here, which is from some work by Mark Chason in this hydatiform mole genome. And it's showing you in the colors here, the red is novel stuff. I'm sorry, the red is deletions, and the darker red is novel, and the black stuff is novel insertions relative to the reference genome. And you can see that for the long stuff, a lot of the deletions have been found already. They were finding a lot of longer insertions. These are actually on 2 different length scales. But you can go all the way down the length spectrum. And here you can see that there's 2 peaks. There's a peak on the right, which is around 6 kb, and these are mostly L1 elements. And in the left-hand plot, there's a peak around 300 base pair, and those are alleles. And you can see that they're also finding a lot of things like VNTRs or STRs at these smaller length scales. And this continues right down really in a continuum down to small indels with this kind of curve that's very pleasing and maybe what people thought was there. But we can really get a look at what is there in a single genome. And the only limitation here is that really we're only able to look at a small set of genomes so far, although it creates a great reference catalog of at least common structural variation and the spectrum of what's out there. And there's just another illustration here of being able to take these finished genomes and then map them back to the reference and find things like a big insertion and to fill in gaps in some cases in the reference genome. So because part of what I want to focus on here in the last part is structural variation and phenotype and especially common disease, one thing we want to think about is how imputable is structural variation. Because even though we are getting more and more sequencing data, we're still just in the low thousands of samples with whole genome sequencing data. And if we're talking about these immaculate genomes, we're down in the tens of samples. So we would like to know how well we can impute structural variation into, because this is MPG, into other larger studies. And probably for this audience, I don't have to talk too much about imputation, but what I'm talking about here is statistical imputation by using the LD that exists in the human genome to fill in missing markers. So if we have a structural variation that we've measured in 1,000 people, and we also have SNP data for those 1,000 people, we can project what the likely copy number states are of that structural variation into a much larger panel of maybe hundreds of thousands of people on which we have array data. But that all depends on how well the structural variants are predicted by the local SNP haplotypes. And the answer is that sometimes they're well predicted by the local SNP haplotypes and sometimes they aren't. In general, the simpler forms of copy number variation, like biallelic deletions or these mobile element insertions, tend to be pretty well imputable. They're pretty well predicted by the SNP haplotypes that they exist on. And this is because they have happened once in human history for the most part. And then like a SNP, right, they're kind of born once, and then they have some trajectory through human evolution. But some of these copy number variants don't seem to completely follow this pattern. So this is an example from HPR, the haptoglobin-related protein. And we know that HPR confers some resistance to certain forms of trypanomyosis. And so one interesting thing about HPR is that it has a very interesting allele frequency. So there's a deletion allele, which you find almost exclusively in Asians. And it's shown on the bottom here. It's a very long deletion allele. It covers HPR and also the HP gene, which is the haptoglobin gene, which is next to it. But you really don't see this deletion in Europeans. And you also don't see duplications in Europeans. But in Africans, you see a segment of the population with a duplication of HPR, and it tends to be a very high copy duplication with a mode around 6 copies. And that's kind of shown on the right. And that duplication really almost exactly BRCAs the HPR gene. So we can look at this as kind of a case study for how imputation works. And this is a little bit of a complicated plot, but it's a plot that we use in our lab a lot. We call them spider plots because of the way they kind of branch out. But the idea behind this plot is that the variant we're interrogating, in this case the the multi-allelic duplication at HPR is in the center of the plot. It's not shown, but it's represented by this white gap in the center of the plot. And then what these colorful bars are that are going out is these are the SNP haplotypes over hundreds of people. So each person's haplotype is one of those branches going out, and the thickness of the branch is telling you how many people are sharing that haplotype across your cohort. And then the vertical bars are showing you the frequency of the SNPs that are on that haplotype background. So it's kind of like a barcode that is labeling each haplotype that a person has. And of course, each person has two of these haplotypes. So this is kind of the overall haplotype structure. So you can see that big thick blue bar going out to the right. That's a very common haplotype going out to the right from HPR. And then there's different frequencies of haplotypes going out to the left. And then these little black ticks that are on the outside, those are telling you the HPR copy number if it's different than 2. So most of the samples here, as you remember from the slide I just showed, are 2 copies. But when you have more than 2 copies, this is showing you where the high copy alleles are. And you can see that almost all the high copy alleles are on this one particular haplotype that's shown at the top. But the other thing you notice is that variation, the 5 versus 6 versus 7 copies, it's also all on the same haplotype. So if you tried to impute HPR, you could tell with some level of statistical accuracy whether or not somebody was high or low, but you're going to have a real hard time telling if they're 5 versus 6, because those are on the same SNP haplotype. So HPR is kind of partially imputable. And what this kind of tends to suggest is that mutational process that's going from 5 to 6 to 7 on that one haplotype is happening faster than the mutational process on the SNP background. So there are no SNPs that are tagging those individual changes because they're happening faster than the rate of evolution of the SNPs.
Is the disease association higher with the multiple haplotypes?
So there is no disease association that we found here. A lot of people have been intrigued by this. It's actually been, this copy number distribution has been known for a while. We stumbled onto it and then realized that other people had seen it, maybe in smaller sample sizes. We haven't found a clear association to phenotype. People have been interested in looking because it certainly feels like there's something going on, that this is very high frequency in Africans. It's probably arisen fairly recently. We don't know. HPR is an interesting gene, but there's, I think, no smoking gun. All right, so we talked a little bit in the beginning about how structural variation, especially really large forms, shows up in rare Mendelian disorders. But I want to spend the rest of our time talking a little bit about whether or not it's also implicated in common disease. And one of the first studies to really show a pretty strong association between a CNV and a common disease was work by Steve McCarroll when he was a postdoc with David Altshuler. And there was a GWAS signal for this gene called IRGM, a GWAS signal from Crohn's disease. But fine mapping had really turned up like no coding variants that seemed explanatory. And then Steve discovered that upstream of IRGM, there was this 20 kb deletion. And it actually was in perfect LD with the lead GWAS SNP. And then Steve went on through some wet lab experiments to show that deletion seemed to mediate the expression level of IRGM1. I think in subsequent years, there's also been some suggestion that there's another SNP that creates an microRNA binding site in IRGM1, and that may also be associated with the regulation. But this was one of the first clear examples where there seemed to be a definite association between one of these large CNVs, that was actually found from array data, and a common disease. Another really striking result came out around 2007 when Jonathan Sabat and colleagues reported that there was this enrichment of large rare CNVs and autism. And it was not so much specific CNVs, but just that cases with autism just seemed to have a higher burden of large CNVs in general, even though they were all over the genome. And so people began to study this, and there became this effort of trying to take all the large CNVs across many cases and say, did they ever happen at the same locus? And some loci began to emerge, like 16p11 was the first one, where there does seem to be some evidence that there's recurrent deletion or duplication on a really large scale at these loci, and that's associated with autism. Today we know there's maybe 8 or more loci where there tends to be evidence of this recurrent duplication or deletion, and then this affects autism. In general, all these large CNVs, they tend to be large. They tend to be like these megabase scale CNVs or close to that, and so they're quite rare in the population, but there is definitely some association, although it explains only a small percentage of autism cases. And even if you take out these 8 loci though, there still seems to be this burden of excess copy number variation in autism cases that's either non-recurrent, in that it's, I don't want to really use the word that it's random across the genome, but if it's not random, the pattern of what it's hitting is not completely obvious, although maybe it's just that we don't have a big enough sample size yet to see that certain pathways are being hit. And there's a similar story actually in schizophrenia, that we see an excess of these rare deletions and duplications in schizophrenia. And similarly, they only explain a small number of the cases. There's some recurrent loci that where you get a fairly good change in odds ratio if you have one of these rare CNVs at these loci. And then there's, again, this evidence for just excess deletions and duplications in kind of a genome-wide way. And most recently, Daniel Hauergrens looked at this in a really large number of schizophrenia samples. And what this is showing is across many, on the right, is across many, many different studies, we're seeing this increase in odds ratio if you have this excess burden of these large, rare CNVs. Although in total, this also still explains a relatively small fraction of schizophrenia. So it's this very interesting pattern in psychiatric disease where this burden of rare CNVs has shown up. And because these are rare, these are mostly the biallelic deletions and duplications. So I want to also talk about some examples that get to these more complex CNVs. And one of the approaches we've used, especially in our lab, to try to understand these complex CNVs, and something that's turned out to be really important and useful, is to start first with the copy number measurements. And then we began to realize that often it gets back to this idea of complexity, where it isn't necessarily just a single CNV. So we would find multiple segments in a region that maybe had different copy number states that weren't all correlated with each other. So we began to look at that in terms of what are the structural haplotypes that underlie these multiple copy number states. And we looked at that both through looking at families and looking at the phasing of the CNVs within families, and also using statistical phasing to try to tease apart the underlying haplotypes. So here is one of the early examples of this kind of approach on chromosome 17. There's a well-known megabase scale inversion on chromosome 17, and there's also regions of copy number variation that are not necessarily correlated with each other. And when we use this technique of sort of deconvolving them into structural haplotypes, we identify 9 different structural haplotypes that are at high frequency in the human population. And really strikingly, 2 of these haplotypes are shown by the red boxes on the right, are at very high frequency in Europeans, but almost absent in the rest of the world. And moreover, these must have gone to high frequency independently. First of all, the history, which is shown by the tree on the left. They're kind of on different branches. And also this large inversion keeps recombination from happening almost all the time in between people who carry either what we call the H1 form or the H2 inverted form. And the other thing Linda discovered was that something that's in common between these 2 haplotypes is they both create fusion genes that involve cancel 1. They're slightly different fusion gene products. And there have been many reported phenotypes in this area. Decode reported that there was that your H1, H2 inversion state relates to fertility in Icelandic women, and there have been a couple other phenotypes reported in here. We don't yet know exactly the significance of the cancel 1 gene, but a lot of people have been following up on this. But this was kind of to illustrate this approach of taking apart these complex loci and trying to figure out what the structural haplotypes are underneath them. Similarly, Christina Usher, a grad student in Steve's lab, worked on the amylase locus. And amylase is striking because if you look at the bottom down here, you can see that amylase exists in up to 15 to to almost 20 copies in different individuals. And it has this other unusual pattern in that most people have an even number of copies of the amylase 1 gene and not an odd number. And this actually derives from the fact that there's this repeating unit that is getting expanded and contracted at amylase, but that repeating unit has 2 copies of amylase 1 in it. So that's why you get this spiky distribution of copy number of amylase 1. And after Christina was able to deconvolve all the common haplotypes at this locus, we also discovered that a finding that had been reported about a year before, that it showed a very strong association between amylase 1 copy number and BMI. Actually, in many other studies where we followed this up, we just couldn't replicate the finding of that association of BMI, probably because a combination of the copy number estimates not being quite so accurate in the original study, and probably some batch effect, even though the investigators tried to control for it, and not understanding the full spectrum of the haplotypes, and especially this sort of odd-even imbalance at amylase. So is this just the tip of the iceberg? I think probably. Our methods for variant detection are improving, and as we get more and more cohort sequenced with whole genome sequencing data, and we do things like deploying PacBio data so we can ascertain more and more variants, it seems likely we're going to find more and more cases of structural variation being involved in disease. And I wanted to end up with sort of another hint of why it's maybe just the tip of the iceberg, and that's that structural variations are actually enriched among EQTLs. So one thing we showed on our study of multi-allelic CNVs was that unlike in plants, there's kind of this direct relationship between copy number and gene expression. We used RNA sequencing data from GeoVetus to look at these multi-allelic forms of copy number variation, like the example I'm showing you on the left to sort of remind you. And then on the right, I'm showing for a gene that's underneath one of these copy numbers, how does gene expression vary with the number of copies in each person? And you can see here that there's almost always this direct relationship between gene expression and copy number. And this isn't necessarily what one would expect. For example, in plants, there's a mechanism where if a plant acquires an extra copy of genes, those extra copies will often be shut off. And the idea is that maybe this is beneficial because you don't want an extra copy to suddenly create an imbalance in the level of expression of a gene. But at least in the human genome, in almost all cases, we find that when there's extra copies of a gene, that expression goes up.
When you say expression goes up, do you mean RNA goes up, or do you mean the amount of protein goes up?
So this was based on RNA sequencing data. So we know that the level of RNA goes up. So we don't have direct measurements of the protein. So also in the most recent 1,000 genome study, we took a look at structural variation and EQTLs. And what the plot on the right is showing here is that these are all the EQTLs, and then we're partitioning them by whether or not the lead EQTL is a SNP or a structural variation. So the red dots, which are mostly above the line, those are places where the lead association for the EQTL is a structural variant. And the blue ones kind of below the line, this is where the lead EQTL is a SNP. And the yellow ones are ones where there's a SNP and a structural variation that are in very tight LD, and it's very hard to tell which one is really driving the association. But the thing is that there's many fewer SVs than SNPs. So if you look at it on a variant by variant basis, in the 1,000 genomes data, they find that SVs are almost 50-fold enriched for being an EQTL compared to a SNP, as long as there's an SV in the vicinity. Altogether, SVs are maybe something like 3% of the EQTLs. But because there are many fewer SVs, this is actually a tremendous enrichment. And similarly, in the GTEx data at ASHG, Colby Chang and Ira Hall were reporting a very similar result. They were finding more like, they had a little bit bigger error bars, but they were finding more like 5 to 25-fold enrichment of SVs for being EQTLs, and accounting to somewhere between 1% and 5% of the EQTLs that they're finding in the GTEx data now across many different tissue types. So this is kind of a hint that when SVs occur, they actually sort of variant for variant tend to have a larger impact on phenotype. And also another thing I'll mention is that in 1,000 genomes, they also found there was a correlation with length. Longer SVs tended to be more likely to be EQTLs and to have more impact on genes. So I hope I've tried to convey to you that I think structural variation is really cool. I think you should think it's cool too. Genome variation occurs at both large and small scales. We need to look at these structural variants as well as SNPs. Although there's fewer SVs, there's evidence that there's actually more impact per variant. They're interesting because they have these faster mutation rates, for example, at the CNVs. And because of that, they're not necessarily always well tagged by SNPs. So it's really important for us to try to ascertain them in addition to just looking at the SNP haplotypes. They may be easier to interpret in some cases because, at least in some cases, the effect is really direct, right? The CNV impacts the exons of a gene, and if you kind of look at the exons as deleting or duplicating, you can kind of predict the direction of effect. And in many cases, that seems to work well. It's certainly not a solved problem. We've got a lot of work to do, especially better ascertainment of these mid-sized variants, a few hundred base pairs up to a kilobase. And also, how best should we go about testing for association to phenotype? So I'd like to say thank you to a lot of my colleagues at the Broad and in the McCarroll lab, and a lot of other people who contributed information for this talk. Thanks very much.